Well, this is a genetics work package which is concerned with trying to determine the relationship between genetic variation which exists between humans and features of aging, such as physical aging and cognitive aging. And the background to that is that we have acquired knowledge over the past 10 to 20 years from animal studies, for instance in flies and worms, those types of animal that have short life cycles and can be studied easily. And we know from those studies that there are specific genes which can dramatically alter the age of survival of the organism. And in parallel with that, over the past 10 years or so, it has become possible to study the human genome in total, so to look at all 3,000 million base pairs of it and to know where all the common variation is, not the rare variation yet, and start to analyse that systematically in something of the same way that has already been done in some simpler organisms. And so putting those two things together, the previous animal studies and the indications that there is genetic impact on all sorts of pathways that matter, and the opportunity to be able to study that now in human is to join those two things together and start to conduct those studies. The principal purpose is to examine a set of common genetic variations which have been well established now to have an impact in some or other disease. So if we move away from thinking about aging and just think for a minute about cancers or heart disease or autoimmune diseases or inflammatory diseases, then there have been identified a number of variations which make a difference to those diseases, but we don't yet know what their full scope of impact is across all sorts of other traits. And for instance, we don't know what their impact might be on physical decline as a feature of aging. So in some ways you could call it measurement science, to take those and then to analyse them in the different contexts that we are here. The main research questions focus on some pathways that have been recognised as potentially being important in aging in other contexts. So, for example, the pathway which is in one of the other work packages concerned with the ends of chromosomes, the telomeres, also has a genetic variation that influences how it works, different in different people. Another example would be the oxidative pathway, so we all brown like an apple as we get old, and so studying variations which may influence rate of oxidation is something that could inform us about things we could change. Our first study, which is currently submitted for publication, was to look at a variation in a gene called TERT, T-E-R-T. In the past 3 or 4 years, it has become clear that there is a variation in that gene which has an effect on risk of all sorts of common cancers. Our second study, which is still ongoing but quite far forward, has been of a variation which is better known for its claimed impact on athletic performance. So it's a gene variation which actually either you have an active or an inactive copy of that gene, and depending on which you've got, your ability to perform to Olympic level in power and sprint sports is thought to be very different from your ability to perform in endurance sports. So one version of the gene is good for one, the other version of the gene is good for the other. We've undertaken a study of that variation in relation mainly to physical capability, and the bottom line there, again very well powered on the scale of this study, is that doesn't make any difference, despite the fact that we know it makes significant differences to what muscle fibre types you have in every skeletal muscle in your body. In the next year, we're scaling up. It has taken us about a year to get our whole system together, because a lot of these data have had to be pulled together from 9 or 10 different places and involving a lot of different people. And we have our system in place now, if you like. And so from those initial studies, we're now in a much better position to be able to analyse quite a much wider range of markers. And so we're shifting gear from our first 1 or 2 through what presently is an additional 10 or so, up to an additional 30 to 50 targets, which have largely been selected and are about to be analysed. And so over the course of the next year, there will be a lot of statistical activity and data sifting surrounding that, which we call our phase 3. The first impact will be in the ways that we develop a greater fundamental understanding of the molecular processes of aging. In terms of the overall strategy, I think one has to think long term, and that's fairly characteristic of the molecular approaches, that you find out what's under the bonnet and then find out how you can change it. And often that may take a decade or more to do, but for some of these things, the inferences could be had that are relevant in the much nearer term. From the angle of impact, the types of variation we're studying and the types of trait we're studying are ones that are relevant to the whole population. So even if the apparent effect in some sense for us might seem disappointingly small, like a 1% or a 0.5% effect in some way, or piece of 1% worth of the information that we'd ultimately like to have, nonetheless, on the scale of your population, that is still a major benefit. And again, one could go back to the cholesterol example and say, well, even if you shift a variation like that in your overall population, it can still have quite a considerable effect on the types of event, like coronary events, that people don't want to have. We can look at it in that way as we analyse these variables. And I guess that's the sort of scale, the type of information we would expect to come out. Small effects, but potentially very informative for a large number of people.